Hello, everyone. My name is Lesley. I am a recent college graduate. I went to California State University, Channel Islands, where I majored in biochemistry and I minored in math. This summer, I was placed in the Salceda lab at the New York Genome Center, and my project is centered around transcriptional evolution in the motor cortex. So just a brief overview of what my presentation will cover. I will talk a little bit about what single-cell RNA sequencing is and how we process that data. We'll go into microglial conservation across species, and then I'll tie that into different cell types in the motor cortex. I want to preface this slide by saying that prior to the start of my internship, I had no idea what single-cell RNA sequencing was. But during my time as an intern, I learned that it's a technique used for sequencing RNA at the single-cell level. And this level of depth allows us to study heterogeneity and cell lineage. And these are two things that you can't really look at with bulk RNA sequencing, because bulk RNA sequencing, generally speaking, looks at average gene expression, whereas with single-cell RNA sequencing, you get a lot more specificity. I also learned how to code in R, which is a common programming language such as Python. And in R, we have a toolkit called Seurat, which allows us to analyze single-cell RNA-seq data. As part of my learning, I went through a lot of tutorials on the Seurat website, and I learned how to do things like clustering and integration, which are two skills that I used for a lot of downstream analysis. So to start my research off, I looked at microglia cells. And microglia are basically cells that function as immune cells in the central nervous system. I started off by reading the article shown on the right, where the main finding is that, generally speaking, across species, there are conserved genes in the microglia that essentially play a role in very cell-specific functions. And despite that conservation, there are also very cell or species-specific genes that can be found. I processed the data sets from this article. I downloaded all the data, which consisted of data from about 9 different species, and each species had several different replicates. I created Seurat objects for the data provided, and I integrated ferret and pig data that I got from the Genome Center. I merged the Seurat objects, I normalized the data, and I integrated that. And then I corrected the batch effect that was present. And something that I also did, but will not show here, is I found genes that were different across species, and I performed gene ontology analysis to figure out what pathways those genes are playing a role in. So this is what my data looks like following integration. The way we would interpret this HuBMAP is basically every spot on the HuBMAP represents a cell, and the cells are clustered according to the algorithm in Seurat, and they are also color-coded, in this case, by species. So on the left is the HuBMAP before the batch correction, and on the right shows the HuBMAP after I corrected the batch effect. And as we can see, because we only looked at one cell type for this analysis, the cells that we see are very homogeneous. And we can't get much information about conservation from just looking at one cell type. So we decided to expand this to look at different cell types in the motor cortex. So the motor cortex is a section in the cerebral cortex involved in the execution of voluntary movements. The article I read for this part of the data analysis was titled "A Transcriptomic and Epigenomic Cell Atlas of the Mouse Primary Motor Cortex," where basically the main goal of the paper was to identify different cell types in the mouse motor cortex and find their respective genetic markers. I processed the data similar to how I did with the microglia data, where I downloaded the data from the article. I then ran SE transform, I integrated the data, and I found genetic markers in each of the Seurat clusters. I identified the top 3 most expressed genes in each cluster, And then using the cell markers found in the article, I made violin plots to visualize in which clusters those genes are enriched. For some clusters that were a little bit harder to annotate, I used Azimuth as another resource where I could find more genetic markers. Additionally, I verified my manual annotations with the predicted/known annotations that were provided in the article as well. And on the right is the HuBMAP showing all the different clusters of all the cells in the data set. And at this point, I had no idea what cell types were present or what cell types corresponded to what clusters. So this is just an example of how I went about annotating the different clusters. On the left-hand side, we'll see a diagram from the motor cortex article, where on the y-axis, we have the different cell types that were found, and on the x-axis, we see all the different markers that were associated with each cell type. And so what I did was I basically took a marker. For example, I used NXPH4 here. I plotted it and saw what cluster contained the highest expression of NXPH4. And because the article listed NXPH4 as a specific marker for level 6 B cells, and NXPH4 was enriched in cluster 9, I could say with relative certainty that cluster 9 corresponds to level 6 B cells. On the left here is the finished and annotated motor cortex HuBMAP with all the different cell types that I was able to identify. And on the right are the different, or is a plot showing how I was able to verify my annotations compared to the annotations that were provided. And as we can see, my annotations align pretty well with the known annotations. So using that, I could say that the mouse data that I have here is a good reference for annotating other motor cortex data sets. And I ended up using that mouse data set as a reference to label the ferret and pig motor cortex data sets that we had in-house. On the left, we have the ferret motor cortex, and on the right, we have the pig motor cortex. And the cell labels transferred pretty well. Once I labeled the motor cortex of, at this point, mouse, ferret, and pig, what I did was I made a list of the cells or the different genes that were conserved between mouse and ferret, mouse and pig, and then ferret and pig. And I created heat maps to get a visualization of how those genes are expressed across different cell types. So the first plots that I'm showing show the conserved genes between mouse and ferret, and how those genes are expressed in mouse and ferret separately. And as we can see, there are some cell types that show very conserved cell markers, such as astrocytes. But once we get into some of the more neuronal cell types, such as level 2 and 3 IT cells, or even level 5 IT cells, there is less specificity. And something else that we can also see here is that the conserved genes show similar patterns of expression across cell types.
2 minutes remaining.
Okay. Here I'm showing the same thing where I took the list of conserved genes between mouse and pig, and I just plotted how they are expressed across different cell types. And again, the expression patterns are similar between the two species. Once I had the list of conserved genes, I ran gene ontology enrichment to see what those genes are doing. And generally speaking, the genes that are conserved are performing cell-specific functions. The last pair that I looked at was ferret and pig. And again, the main takeaway here is that genes that are conserved show similar patterns of expression across cell types. And again, the genes that are conserved are performing cell-specific functions. In addition to looking at genes that are conserved, I also looked at genes that were expressed differently across cell types. And the first example that I'm highlighting here is NXPH4, which, if you recall from earlier, is a marker for level 6 B cells in the mouse motor cortex. And as we can see here on these violin plots in the middle, it is very specific for level 6 B cells, but it is not expressed at all in the ferret. and in the pig, the expression is nonspecific and is scattered across different cell types. Another gene that I looked at was MBP, and sort of contrary to the previous slide, it is very specific in the ferret and nonspecific in the mouse and the pig. The last example I wanted to look at was APOE, which is known as a canonical marker for astrocytes. And as we can see, it's not expressed very highly in ferret, nonspecific in pig, and nonspecific in mouse. So some of the conclusions that I was able to draw from this analysis is that genes that are conserved across species carry out cell-specific functions, such as calcium binding and ion binding in excitatory cells. Having a good reference, such as the mouse data set, enables cross-species data analysis. And some canonical cell markers, such as APOE, may not be conserved across species or may not be specific. Some of the next steps that I would take if I were to continue this project was looking more deeply at cell markers that are not conserved and seeing if those cell markers are expressed differently in different regions of the brain, such as the hippocampus. And finally, I just want to acknowledge my mentors, Saket, Avi, and John, for teaching me so much in so little time. Everybody at the Salceda lab at the New York Genome Center for being so welcoming, and the NIH HuBMAP Consortium for giving me this opportunity to do research and to learn so much from so many incredible scientists. So thank you.